Okay, welcome everybody. So this is the lecture on Genome Organisation for the Molecular Genetics module, the Biomedical Sciences programme at the University of Bradford. So we're going to be talking today about the organisation of eukaryotic genomes. We will pay reference to prokaryotic genomes, but only as a comparison really to what's going on in eukaryotic cells. What we have here is an electron micrograph of an E. coli cell spilling out its nucleoid, its genetic material. So you can see here that there's quite a lot of the DNA, more than you may perhaps imagine, compacted within these bacterial cells. But if we could compare that to an electron micrograph here of a eukaryotic chromosome, what you can see immediately is far more material here, just in a single chromosome, than is present in the entire prokaryotic genome. So we need very, very, very complex and careful organisation of all this DNA in order to be able to, one, package it all inside a eukaryotic cell, and two, actually access it when we need to in order to replicate it and use it to make messenger RNA and protein. So the lecture today is going to be talking about two things really. The first thing we're going to be talking about is the different types of genetic information stored within eukaryotic genomes, and the second thing is basically how exactly that information is being stored and how that storage is regulated in order to enable us to access the DNA for transcription and DNA replication. So there are two excellent chapters available for additional reading for this topic in the core textbooks. There's an excellent chapter in the Brooker textbook and also an excellent chapter in the Molecular Biology of the Gene textbook, which deals with genome organisation and the sorts of things we're going to be talking about in the lecture today. So just a quick overview then of what we're going to be discussing. We're going to start by talking about variation in the genome itself, and in particular variation in genome organisation. And that'll lead on to a discussion about the types of information that's actually stored in eukaryotic genomes, because as you'll see, it's very different in nature to what's present in simpler prokaryotic genomes and those simpler organisms such as bacteria. We'll then talk about key elements within the eukaryotic genome itself, paying reference really to chromosome structure and key elements within chromosomes, before moving on to basically a discussion about how we build a chromosome in a eukaryotic cell. It's a stepwise process that involves lots of different proteins and different types of regulation, and we'll touch upon some of those in this lecture, and then finish the lecture really by talking about how we have to manipulate those same structures in order to gain access to DNA so that we can replicate and transcribe it, or we being the cell obviously when it needs to. Okay, so we'll begin with some of the basics, just a very brief refresher really. Sure, most of you will know this already, but as you all know, prokaryotic genomes are very simple. Often bacteria have a single chromosome that exists as a circular nucleoid, and it exists within the prokaryotic cell in a non-partition manner. In other words, it just kind of floats around inside the nucleoplasm. Inside the protoplasm. It floats around inside the cytoplasm within the prokaryotic cell. This is in direct contrast to eukaryotes. One of the defining features of a eukaryote being the presence of a membrane-bound nucleus, which is used to partition the nuclear material, the chromosomes, away from the cytoplasm where protein production takes place. Eukaryotes often have more than one chromosome. Some eukaryotes have many chromosomes. Some only have a few, like yeast. But generally, these chromosomes are much larger than the prokaryotic counterparts. If we think about genome size, you'd be forgiven perhaps at first thought to thinking that the larger a genome, the more complex an organism. It's kind of an obvious comparison, and certainly there are some obvious correlations between genome size and complexity of organisms. So if we look here at two very important organisms in biology, E. coli and Homo sapiens, us. If we look at the size of the genomes in E. coli, 4.6 megabases versus 3,200 megabases for Homo sapiens. No great surprises there. It seems obvious, and of course it is obvious, that we would need a much larger genome in order to generate such a complex multicellular organism as humans. Yeah. However, it's not quite as straightforward and simple as size, because if we look at the genome sizes in some other organisms, such as locusts, for example, and tulips, which is this second organism here, we will see that these organisms, whilst obviously nowhere near as complex as humans, both have genomes in excess of the size of the human genome, and in the case of tulips, the genome size is huge, 120,000 megabases. So it's not simply a case of bigger is better. It's not the size that matters, but what actually you're doing with that genomic real estate. So what do I mean by that? Well, if we take a look at something else that's really interesting, something called gene density, you'll start to see how this is unpicked. So gene density refers to the number of genes present per megabase of DNA. So if you look at a megabase of the E. coli genome, how many genes are found within that region? This table here illustrates those points. So if we look at E. coli, we see that there's around 950 genes for every megabase of its genomic DNA. Now if we compare that to a more complex organism, let's say Drosophila, we see now that Drosophila, in contrast to E. coli, only has around 82 genes for every megabase of DNA in its genome. So the number of genes present is far lower, the gene density is far less. If we take this to the ultimate extreme and look at humans, which have the lowest gene density of all these listed organisms, we see that we have around 6 genes per megabase of DNA. So an incredibly low gene density. So gene density is inversely correlated with the complexity of an organism, and it's a really, really good correlation that stands up to interrogation. This. So what's going on then? Because clearly we've got lots of genomic real estate with very large genomes, but very few genes. So given that, what's occupying all this space? So this figure illustrates nicely what's going on. So if we look here, we've got E. coli, Cerevisiae, Drosophila, and then humans. And as you can see, they've got all these different color-coded regions within this 60,000 base pair segment of the genome. Now, E. coli, if you look at that, 60,000 base pairs, almost all of the information, all of it is comprised of protein coding genes. There's some very small regions of intergenic sequence, which is just coding for nothing, or regulatory elements, but the vast majority of this genome is encoding protein coding genes. Ignore the red region, that's still a protein coding gene. It's just one that's been highlighted for illustrative purposes, as I'll talk about in a second. If we go to the next organism, then Saccharomyces cerevisiae, brewer's yeast, also used to make bread, baker's yeast, we see that the actual structure of the Cerevisiae genome has a lot more in common with prokaryotes than it does with humans. Saccharomyces cerevisiae is a single cell organism, it's a simplest eukaryote, and it's also got a very high gene density, so the vast majority of the Cerevisiae genome is comprised of protein coding genes. The keen-eyed of you may have noticed that in addition to all the green, we do have some small regions here of blue, and what these blue regions represent are so-called intronic sequence. Introns represent the fact that, as we'll discuss later on in the module when we talk about gene expression, eukaryotic genes are comprised of coding exonic regions and non-coding intronic regions. And these intronic regions are removed prior to protein translation and following transcription by a process called splicing. Now, the presence of introns and intronic sequence in Cerevisiae is very, very rare compared to more complex eukaryotes, but it does have some, as illustrated in this figure here. Now, if we go on to an invertebrate now, a much more complex multicellular organism, you can see now that the genomic picture is looking incredibly different. We've gone from the majority of the region of this 60,000 base pairs being comprised of protein coding genes to the green actually being more or less in the minority. There are much, much more prevalent now intronic sequences, and these intronic sequences are getting larger. We still have these intergenic sequences that are coding for nothing, and we also now start to have a really important type of sequence called repeat sequence, which is indicated in yellow here. So if we move now onto our genome, 60,000 base pairs, normally we would get 2 genes spanning this sort of distance, in contrast to almost 60 genes in E. coli. And what we can see now, interestingly, is there's been again another really large leap up in the percentage of the information contained here that's comprised of these repeat sequences. So our genome is littered with repeat sequences, as we'll discuss in a second. You'll also notice that this red gene, which was highlighted in E. coli, the RNA polymerase gene, now spans a much, much larger area of the genome than it does for E. coli. And the reason for that is that the vast majority of that genetic space is occupied and taken up by either intronic sequence or repeat sequence. The minority of the sequence is the sequence which codes for protein. So if you look at this example here, all of these little areas of red represent the coding sequence regions, and it's the areas of blue that are taking up the majority of the space, i.e. the intronic non-coding sequence. So there's a very different picture when you look at different types of organisms and the types of information contained within their genome. So there's a very strong correlation, as we've said, between organism complexity and low gene density, and there's also actually a really strong correlation between organism complexity and the size and the amount of intronic DNA present within those genomes. So it looks on that previous picture as though eukaryotic, or if you like, human genes are much larger than prokaryotic genes or the genes of simple eukaryotes, and that's quite frankly just not true. So the reason why it looks as though the genes are larger is simply because they're spread out over a much bigger area. The protein coding sequence in eukaryotic genes, as I've mentioned, is usually discontinuous, and depending on the complexity of the organism, the amount of discontinuous or intronic information increases. So here we have another example of a correlation between organism complexity and gene structure in this table here. This time looking at the average number of introns present per gene. So E. coli prokaryotes don't encode any intronic information. They simply do not want to waste that genomic real estate on non-coding intronic sequence. Similarly, Saccharomyces cerevisiae, very few introns, as we saw in that previous cartoon, just 1 or 2 areas of blue in that 60,000 base pair region. So per gene on average 0.04 introns. So in other words, of the 4, 5, 6,000 cerevisiae genes, only a handful, only 1 or 2 genes actually do contain an intron. The situation starts to become completely different when you start looking at more complex organisms, including invertebrates and mammals, where we start now to see many introns per gene. So the average number of introns per gene in humans is around 6, although some genes have many more than 6. So, for example, cystic fibrosis transmembrane regulator is comprised of 23 different exons, if memory serves. So a much larger number of discontinuous introns found within that particular gene. So why have introns at all? What's the purpose of spreading your gene out over the genome? It seems counterintuitive and just a waste of time and genetic space, really. What purpose do these introns serve? Well, they actually serve a very important purpose. By distributing their information in a discontinuous fashion, what complex eukaryotic cells are able to do is generate many different isoforms of a protein from a single gene by mixing up the exonic sequence in different ways to produce what are known as alternative splice products. So we'll talk a little bit about alternative splicing when we talk about gene expression later in the module, but this is a topic which we'll cover in quite a lot of detail next year on medical genetics. So if you recall, one of the other areas that was really prevalent in more complex organisms and their genomes was so-called intergenic DNA. So intergenic DNA doesn't code for proteins or structural RNAs at all. It's the information found between genes, if you like. And again, more complex eukaryotes seem to possess much more of this intergenic DNA sequence. And up until quite recently, really, the past 10-15 years, a lot of people perceive this intergenic DNA as junk DNA. And it used to be referred to that in textbooks, actually, up until quite recently. However, we now have a much better idea of what this junk DNA, this intergenic DNA, is comprised of, and interestingly, it appears to actually be very important, both in terms of regulating the genome and in terms of conveying complexity to the organisms which possess it. So we can broadly categorize intergenic DNA into 2 different groups: functional stroke non-functional intergenic DNA and repeat sequences. So we've already seen how repeat sequences really start to explode into genomes as you get into more complex eukaryotic organisms, particularly into humans and into Homo sapiens. If you look at our entire genomic sequence, so 3,200 megabases, around half of that sequence is actually made up of repeat DNA sequence. So that's a staggeringly large amount of actual repeat sequence, taking up all that kind of space and real estate within our genomes. Now, this repeat sequence itself is comprised of 2 different types: microsatellite DNA and genome-wide repeats. Microsatellite DNA represent replication errors, and they tend to be dinucleotide sequences, runs of C's and A's, C's and A's, C's and A's next to one another. Now, the reason these microsatellites appear within our DNA is that runs of dinucleotide sequences such as that type, CA, 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 etc., are very tricky to replicate for the cell. And what can happen during DNA replication is a replication error, whereby you have slippage of the replication fork, which leads to an increase in the actual size of that microsatellite repeat. So it's not a common event during replication, it's quite dangerous to the cell, but over many years, obviously, of evolution, these microsatellite DNA repeat sequences have kind of embedded themselves within our genomes. The other type of repeat are known as genome-wide repeats, and these represent transposition events. So we're going to be talking about transposons and transposition events in more detail later on in the module. When we talk about homologous recombination and transposons. But essentially these are ancient repeat sequences of DNA which again over evolution have managed to expand and occupy, take over like weeds really, a lot of our genomic real estate. Now transposition events within human cells and in mammalian cells is a rare event, it doesn't happen a lot. That's one of the reasons for that is it's very dangerous, it generates a lot of genetic instability and can lead to mutation, but nevertheless these sequences have been around for so long that they've kind of co-evolved with our genomes and gradually over time just come to occupy a lot of the space within them. If we have a look at this figure here, what this is doing is trying to break down the human genome into these different types of sequences, either gene or gene-related sequences such as introns and exons, etc., which occupy maybe about 1200 megabases, and then intergenic DNA, which is a type of DNA we've just been talking about, which include these microsatellites due to replication error, genome-wide repeats such as a transposition events, but also as well in some cases unique regulatory regions which include things called microRNAs and long non-coding RNAs, which are a new type of regulatory RNA that we'll be discussing in a lecture later on in the semester. So we've arrived now at this realization that much of this intergenic DNA presence far from junk. As I've just explained, it's comprised of lots of different elements, but you can kind of arrange them functionally into two broad types, and those are transcriptional regulators and non-coding RNA. So transcriptional regulators represent the enhancer and insulator sequences found upstream of genes, which are used by cellular transcription machinery in order to either enhance or dampen down expression of genes during transcriptional programs. And non-coding RNA, including microRNAs, which we've just discussed, but also long non-coding RNAs as well. In fact, even regions of the genome which were long thought really to represent just dead genes or pseudogenes, to give them the proper name, are now being shown to have some level of function. So certain pseudogenes have been shown to be transcribed to produce long non-coding RNAs or other elements that themselves can then impart a function onto transcriptional programs in the cell. And if we think about the Human Genome Project as being the collaborative project to try and sequence or to sequence the human genome in its entirety, we have now got another project called the ENCODE project, which was recently completed, which had a much bolder aim in many ways, and their aim was to identify all the functional elements present within the human genome. Because following the completion of the Human Genome Project, it became pretty quickly evident that there weren't that many genes present actually in the human genome. Around 22,000 genes is a conservative estimate of how many genes we encode. Nowhere near the number that was originally postulated, and we've realized now that we can get away with so few genes because of how complex the systems are within our genome for the controlled expression of those 22,000 genes. And the ENCODE project set about the business of trying to identify all the nuances of the genome which lend itself towards controlling gene expression profiles, and that includes these non-coding RNA elements, these transcriptional regulators, and indeed epigenetic changes to the DNA and associated proteins, which offer another level of regulation in terms of gene expression control. So in contrast to prokaryotic organisms whose genomes are circular in nature generally, eukaryotic genomes are generally stored within these long linear DNA structures termed chromosomes. So I'm sure many of you will be familiar with this image here of classical metaphase spread of human chromosomes, in which you can see some of the major structures found within human chromosomes, including the centromere, which is located centre of the chromosome here, where the chromatids intersect, and the telomeres, which are located towards the ends of these linear chromosomes. The other major structural feature of a chromosome being origins of replications. And we'll talk more about origin of replication and telomeres when we discuss DNA replication. Origins of replication are sites at which DNA replication initiates, and for human chromosomes there are many origins of replication across each chromosome, in contrast to prokaryotic genomes, which generally have a single origin of replication. And telomeres, which are very important structures within these linear chromosomes, as they mask the end of the chromosome, the end of the double-stranded DNA, if you like, from machinery within the cell, which would otherwise recognise it as a double-strand break. And as you'll be aware from the lecture on DNA damage and repair, double-strand breaks within cells are real red flags to the DNA repair machinery. So the telomere is a specialised structure which enables the chromosomes to mask, if you like, their linear end from that machinery and make sure that DNA repair enzymes don't come along and start trying to join chromosomes together, which obviously would be very bad for the cell. Centromeres then, which we're not going to discuss in the DNA replication lecture, so I want to cover in a bit more detail here, are another really important structure found within chromosomes. And they're generally found, well, in the case of an acrocentric chromosome, at the centre of the chromosomal region. Very broadly speaking, they're important because they're the site of the chromosome which the cell latches onto when it wishes to move the chromosome around inside the nucleus. So the most obvious time when this happens is during mitosis or meiosis, when chromosomes are replicated and then need to be separated into each daughter cell prior to cytokinesis, cell division. So to do this, the cell latches onto the centromeric region by virtue of a series of proteins known as kinetochore proteins, and it latches on with tubulin, which is a structural microfilament found within the cells that forms part of the actin and tubulin cytoskeleton. Centromeres themselves are quite interesting in terms of the structure. They differ between eukaryotic cells. For example, Saccharomyces cerevisiae has a very ordered type of DNA sequence which is located at the centromeric regions, and this is kind of the odd one out, as if you compare that to other yeasts, Schizosaccharomyces pombe, which is a fission yeast, or indeed more complex organisms such as Drosophila or Homo sapiens, what we find in these organisms is that there isn't a defined genetic sequence. Rather, the centromere is primarily comprised of repeat DNA sequence, and this can vary in size depending on the organism which you are looking at. Okay, so we've looked a little bit there about some of the different elements that exist within eukaryotic genomes, and also we've taken a quick look at some of the major structural elements of a chromosome. But how do you actually go from eukaryotic DNA to a eukaryotic chromosome? What are the processes and steps required to build that structure? So that's what we're going to focus on for the remainder of the lecture. And as you'll see, it's a multi-step and quite complex process that ultimately involves an ordered association between DNA and protein. So chromatin, which is what we are looking at when we look at this sort of material here in a chromosome, or indeed this sort of material here, which is a magnified chromosome through an interface, isn't DNA, but rather the association of DNA with a particular group of proteins called histones. So as we zoom in on these different chromosomal structures, what you will see, if you look at higher magnification, are two different sorts of high order structure. The first is what's known as a 30 nanometer fibre, also sometimes called a solenoid. And then if you look elsewhere, you also see a smaller structure known as a 10 nanometer fibre. And it's this 10 nanometer fibre which is also being referred to as the so-called beads on a string formation, which you can appreciate if you look carefully at this structure here. So the beads on a string formation was some indication, at least, that DNA is associating with some other protein structure in order to order its assembly into chromosomes. So what exactly is that other protein, and How is it functioning? So eukaryotic DNA associates with a conserved family of small proteins known as histones, and together the association of DNA with these histone proteins form structures which are known as nucleosomes. So the nucleosomes in the beads on a string image I showed you in the previous slide would represent the beads, and they are in fact disc-like structures comprised of 4 histone proteins around which the DNA wraps itself. So each nucleosome consists of around 147 base pairs of DNA, which is wrapped around the nucleosome 1.65 times. The nucleosome itself, as I've said, is comprised of histones, 4 different histone proteins which associate as dimers to form what's known as a histone octamer. So 4 different proteins which exist as a dimer, 4 of those dimers come together to form a histone octamer. So 8 proteins in total, 4 pairs of histones. The histones which go into the nucleosome are histone 2A, histone 2B, histone 3 and histone 4. Histone 2A and histone 2B form a dimer with each other, and 2 of those histone 2A, histone 2B dimers are comprised in the nucleosome, and the histone 3 and histone 4 proteins form a histone 3,4 tetramer, which makes up the other half of the nucleosome around which the DNA wraps. Now there is a 5th histone protein, it's called histone H1, and histone H1 isn't part of the actual histone octamer itself, so it isn't part of this structure here around which the DNA wraps. Rather, it localizes to a region between these nucleosome structures, which is known as a linker DNA. And as we'll see, histone H1 has a very important role to play in the regulation of DNA with histone octamers. So it's able to manipulate how these histone octamers and DNA are interacting, which is an important ability that the cell needs to make use of during DNA replication and transcription. So the first person to propose the nucleosome model for DNA histone interaction was Roger Kornberg. And a guy called Knoll tested Kornberg's model by use of an enzyme called micrococcal nuclease, or MNase. Now, the primary property which determines whether or not micrococcal nuclease is able to degrade the DNA, cleave the DNA to which it's bound, is whether or not that DNA itself is associated with proteins. So, for example, if you have naked DNA, which isn't associated with any protein, then micrococcal nuclease will degrade that DNA very readily. If, however, that DNA is interacting with protein, the nuclease is unable to access the DNA correctly and will not cut the DNA. So Knoll hypothesized that by using micrococcal nuclease, he could test whether or not DNA was being wrapped around these histone proteins in the ordered manner that Kornberg was hypothesizing. And when he did his experiment, he observed exactly what Kornberg had put forward. So he observed that different sizes of histone-associated DNA were observed following the experiment, depending on the duration that the MNase enzyme was incubated with the sample. So, for example, if you did a relatively short incubation with DNA, with chromatin and micrococcal nuclease, and then you ran an agarose gel and looked at the different size DNA fragments being produced during that experiment, you would see that you would generate a ladder of different sized DNA fragments, each of which, broadly speaking, differ by around 200 nucleotides in length. And what this represented was a micrococcal nuclease cutting in between the resident histone proteins which are present around which the DNA is wrapped. So if you do a relatively short experiment, what you see is the micrococcal nuclease. Apologies, my computer's just is just locking up, hence me pausing and trying to sort it out. So if you do a short experiment where you incubate DNA wrapped around nucleosomes and micrococcal nuclease, that enzyme will cut the DNA in between those nucleosome proteins. And it won't always, with a very short incubation, cut every time between the nucleosomes. It might cut every other time, or in between every 3 or 4 nucleosomes. And that's why you see this banding pattern in terms of DNA fragment. It's why every single fragment isn't 200 base pairs, because the enzyme hasn't cut between every single nucleosome. I've cut every other nucleosome, or every 4 or 5 nucleosomes, but ultimately it generates a ladder of different fragment sizes. Now, if you leave micrococcal nuclease long enough, it will efficiently chew up all the DNA that isn't tightly associated with protein. So that's what Noel did. He also did a long, extensive digestion. And when he left his samples for a long time and then looked at what DNA was present, he found that there was a single species of DNA, 147 base pairs in size, that was left after digestion with the enzyme. So what this told him was that there is a region of DNA around 147 nucleotides long that is tightly associated with the nucleosomes and therefore permanently non-degradable by MNase digestion. And that represents the 1.65 rotations round the histone octamer that we know DNA makes when it forms these nucleosome structures. So together these experiments confirmed Kornberg's hypothesis. Okay, so one interesting fact about the histone-DNA interaction is that it occurs in a sequence-independent manner. So it'd be very inefficient of the cell to try and evolve proteins to interact with DNA based on sequence. So rather, the histones have developed an interaction or possessed a strong interaction with DNA based upon their amino acid sequence, and in particular the fact that they're comprised of a large number of positively charged amino acids, arginines and lysines. And it's these positively charged regions which interact with the negatively charged DNA. One of the consequences of this interaction is that DNA is forced to bend around the histone octamer, and it achieves this via an interaction with a minor groove of the DNA via a series of hydrogen bonds. This bending of the DNA is assisted by the fact that there's a big polar difference between the histone proteins and the DNA itself. So the histone proteins are very positively charged, whereas the DNA is very negatively charged, which again aids in this kind of bending of the DNA around that histone octamer disc. An important consequence of the DNA wrapping around the histones in this manner is that the histone tails, the N-terminal region of the histone proteins, ends up in the mature nucleosome. These tail regions end up actually emerging from the histone octamer structure, from the nucleosome, in an ordered manner. And again, this process is important because it's another way of assisting the DNA to wrap around the octamer in the correct orientation, which is in a left-handed formation. Now you might ask, well, why on earth is it so important that the DNA wraps around the histone octamer in such a precise and ordered way? Well, one of the advantages of wrapping the DNA around the histone in this manner is that it generates something called negative superhelicity. And this negative superhelicity or tension in the DNA as it wraps around the histones enables more efficient DNA replication and transcription when the time comes to unwrap the DNA from these nucleosomes. It's got stored energy and stored negative superhelicity, which enables that process to occur more efficiently. So we've talked about how the histone proteins in the DNA interact with one another, how the DNA wraps around these histone octamers. Well, what happens once you've got DNA wrapped around the histones, once you've formed this beads on the string structure? Well, the next thing to happen then is the recruitment of this fifth histone protein, histone H1. So when histone H1 is recruited to the linker region of the nucleosomes, it functions to ratchet and tighten up that entire structure, as depicted in this cartoon here. Now the tightening up of this histone structure leads to the formation then of the so-called 30 nanometer fibre. So we go from this beads on a string formation here into this higher ordered, larger 30 nanometer structure. From this 30 nanometer structure we then end up forming what are known as chromosome scaffolds by the interaction of these 30 nanometer fibers with a whole different range of chromosomal structural proteins called SMN proteins, and then together the SMN proteins form, along with the 30 nanometer fibers, the sorts of structures we saw a few slides ago, and on the first slide of the lecture, where you have these loops of chromatin associated with the central proteinaceous scaffold of the chromosome. So we go from naked DNA, which wraps around histone-octamers. The histone-octamers then themselves form a higher order structure known as a 30 nanometer fiber, and that process is driven in part by the recruitment and action of histone H1. And the 30 nanometer fiber then goes on to form the higher order chromosome structures that we would see by electron microscopy. One of the consequences of forming the 30 nanometer fiber is, of course, that your DNA becomes even less accessible than it was previously when it was wrapped around the nucleosome. So when you have 30 nanometer fiber formed, what you've got is very, very transcriptionally silent and latent DNA that isn't really doing anything in the cell. And one of the big complexities around eukaryotic and higher order chromosomes is how this structure is unraveled, how the DNA is released from this kind of constraint so that the transcriptional and replication machinery can access it and express the genes or replicate the DNA. It's clear that 30 nanometer fiber formation itself is dependent upon intact histone tails. So we mentioned the histone tails before. The histone tails are actually one of the most interesting parts of the histone proteins. So we talked about how the histone tails help in the previous slide the DNA wrap around the nucleosome in the correct way, in this left-handed manner, to generate negative superhelicity. However, the histone tails are very, very important in other aspects as well. So the histone tail regions, you can imagine them as kind of like fingers emerging from these very inert nucleosome structures, fingers of proteins, of peptides, which the cell can modify post-translationally by phosphorylation, by methylation and acetylation, and all sorts of other post-translational modifications in order to regulate how the histone proteins then are holding onto the DNA. A modification of histone tails is a really, really important area of genetics and biology in its own right, and something that you'll learn a lot more about in medical genetics next year from Dr. Fessing. In this instance, we know that the actual tails are also important upon the formation of for the formation of the 30 nanometer fiber. Now, one of the interesting consequences of this higher order structure, these 30 nanometer fibers becoming tethered to these chromosomal and nuclear scaffold proteins, these SMC proteins, is the formation of what are known as chromosome territories. And this is a concept that chromosomes don't just exist within the nucleus in a random manner. Rather, the chromosomes themselves have particular regions and domains or territories within the nucleus where they're most often found. Now, why would this be important? Well, we're now beginning to understand that transcriptional control at some level, at a certain top level, is impacted by these chromosome territories. And what we can see actually is that groups of genes known to function in similar pathways, such as epidermal differentiation and things like that, tend to congregate within the same region of the nucleus. And work goes on here at Bradford actually looking at 3D nucleus spatial structures and differentiation in skin to try and determine how and why chromosomal regions are localizing to these specific areas of the nucleus in order to more efficiently, we presume, engage genes involved in that particular process in different skin differentiation. So I mentioned a couple of times that it's really important to realise that nucleosomes are dynamic entities. So once you've wrapped your DNA around a histone-octamer, it's not static. It isn't like that forever. If that was the case, we'd never be able to replicate as DNA, and we'd never be able to transcribe our DNA and express genes. So even though there's a spatial need to, if you like, constrain and wrap our DNA around all these different proteins to form these quite complex higher order structures, there's an equal need to be able to unwrap them and to give access to the DNA. So how is that achieved? Well, one of the ways in which it's achieved is by sliding the DNA or the nucleosome along sliding the nucleosome along the DNA to reveal particular regions of interest. So if you think about the cartoon example here, if we have two different proteins that wish to bind a particular region of the DNA, and we can see that those currently in this structure here where the DNA is wrapped around the nucleosome, those protein binding sites in the DNA are masked. So in the same way that micrococcal nucleases wouldn't be able to digest that DNA, these protein interaction partners can't bind to that DNA region either because the interaction remains blocked by the presence of the nucleosome. Well, we can slide the DNA, slide the nucleosome along, so slide that bead along the string so that it reveals a binding location for protein 1. And if the binding location is relatively close to the end of the DNA that's wrapped around the nucleosome, that might be quite a frequent event. It might be quite a straightforward event for the cell to undergo. What might be much more unusual is then the binding of a second protein that requires much bigger migration of the nucleosome along the DNA strand. So these, if you like, modifications, these movements of histones, again, involve nucleosome remodeling, which is a process which can be driven by ATP, or it can be ATP independent, and crucially also involve the post-translational modification of the histone tails, the sorts of modifications we were talking about earlier on. And as I mentioned, you'll learn a lot more about these processes in medical genetics next year. Finally, if we consider DNA replication and think about the replication fork moving along the DNA, clearly when the replication fork meets a histone octamer, it needs to be able to get rid of that histone octamer, replicate the DNA, and then the histones need to reassemble behind the newly replicated DNA as the fork moves away. What's really interesting is that you get different combinations of the dimers that form these histone octamers reassembling with the DNA following replication, in such a way that the old parental nucleosome gives rise to different proportions of the new nucleosomes, which are then used to wrap the DNA around following DNA replication. What this means is that there's an actual possibility of inheriting nucleosome modifications following DNA replication, so there's a kind of epigenetic, if you like, memory that's possible in terms of inheriting modified histones in a cell which has just undergone DNA replication. And this histone code is one of the ways in which we think there's an added level of complexity to how our DNA is used during transcriptional programs in cells. So just to summarise what we've talked about in the lecture, so we've gone through things broadly speaking here, and as I said, there's excellent chapters in both recommended textbooks. But genomic DNA in eukaryotes clearly contains a diverse amount of information. So it's not just genes in higher order eukaryotes. Our genetic information is comprised of genes, but also intergenic regions, which contain regulatory DNA and also encode for regulatory RNA molecules, which we'll talk about later in the module. A lot of the information found in higher order organisms is actually functional in nature, so it might look like junk, or it was considered junk historically, but we now know that it's actually this so-called junk information which is often regulating the protein coding genes. Chromosomes, or chromatin, is effectively comprised of DNA and histone proteins, insofar as the DNA wraps around histone octamers to form these nucleosome structures, which then themselves form these higher order structures such as the 30 nanometer fibre. This is required for two reasons, well, for several reasons actually. It's required for the efficient packaging of information. So if we didn't organise our DNA around nucleosomes, we simply wouldn't be able to fit it into the nuclei of cells. So we have to have a higher order packaging system, and this is what it is, this nucleosome structure. It also allows a regulated access to the information. So one of the disadvantages of wrapping DNA around nucleosomes is you make it inaccessible, but that is also an advantage actually, because it means we can regulate that access, and by regulating access we can do more complex things with the genetic information found within our genomes, and thus generate more complex cells and organisms. And there's also the possibility of enabling a phenomenon known as the inheritance of genetic and epigenetic memory. So this passing on of parental histones, which may themselves be modified epigenetically and post-translationally to newly replicated DNA in daughter cells. Okay, so I hope you found this overview of the Genome Organisation lecture useful. Be sure to check out the other lectures on this playlist, and always remember, that the core textbooks represent an excellent source of information for which to supplement these lecture slides and these podcasts. Thank you.